In muscle physiology, you will often hear something known as EC coupling, or what is known as excitation contraction coupling. Okay, and so there is a stepwise pathway in order for things to take place, right? So if we graph a little bit of stuff over time, we're going to graph some things over time. And the first thing that we're going to graph over time is we are going to graph membrane potential in millivolts, right? So if in blue, right, let's graph, we have a resting membrane potential for a motor neuron. This is going to be in the motor neuron, right? Now remember, we send that signal in the motor neuron, and it goes through these stages of an action potential, like so. So notice that here we start this signal, right? But then it's here where we generate the action potential. And then the action potential is generated in the motor neuron, propagates all the way down the motor neuron, and then you have the flow of information between the motor neuron and the muscle cell at the neuromuscular junction. So now if I graph the membrane potential of a muscle cell, right, it's going to look something like this. Okay, so notice that I first sent my signal here, right, with the signal starting in the motor neuron, generated an action potential in the motor neuron here, and then generated an action potential in my muscle cell here. So there's a little bit of time in between when you have the activation of the action potential in the motor neuron and the activation of the action potential in the muscle cell. And this is about 2 milliseconds. Okay, so 2 milliseconds. Now, if we add in a third graph, and this third graph is going to be tension. Okay, and this is going to be tension of the muscle cell. So what's going to happen is that you have all of that stuff happen, right? And then all of a sudden the tension is going to go up as the muscle cell starts to contract. Going to hit that tension, and then it's going to come down, and it's going to come back to rest. So this right here, right, that's your period of contraction. Right? And then from here to here, that's your period of relaxation. Okay, but notice that from the time that the action potential was generated in the motor neuron to the time at which you get sort of this uptick of the tension, that contraction, right, there's a delay in that time. So that delay is what we call the latency period. And that latency period is really just the amount of time that is necessary for all of those events that we talked about, right? Generation of the action potential in the motor neuron, propagation down the motor neuron, generation of the action potential in the motor end plate, propagation in that, traveling down the T-tubule, activating the foot proteins to release calcium from the sarcoplasmic reticulum, calcium binding to troponin, troponin causing a conformational change in tropomyosin, it shifting, and then myosin binding, and then going through the hydrolyzation process to get to the power stroke. All of that takes time, right? And that all happens during the latency period. So it's this point here, right? That's your first sort of power stroke. Okay, so the time of the initial signal to the time of the first power stroke, that's what we refer to as the latency period. So if you look at the phases of muscle contraction, you'll have the latency period, period of contraction, and then the period of relaxation. Okay, now if we graph muscle contraction, and I'm going to slide this way over here because I want a lot of room in order to do this, right? If we do this long big graph, and we're going to put tension over here, okay? And what I want to do is I want to have zero tension here just to give me a bit of room. Okay, so we're going to set 0 at that point right there. Okay, so what's going to happen is what happens if I send an action potential, generate an action potential in a motor neuron. What I'm going to get is what we call a muscle twitch. Okay, so it goes up, goes up, contracts, relaxes, returns to minimal tension. What happens if I send another signal? Same thing. It's going to go up, going to come down, and it's going to go back to the relaxed state. Send another signal, same thing's going to happen. It's going to go up, it's going to come down, it's going to go back to its relaxed state. So what happens if I send a signal, muscle comes up, starts to contract, then it starts to relax, but before it relaxes, I send another signal. What's going to happen is that you're going to get a secondary twitch, like so. And if I send another signal before it relaxes, I'm going to get another twitch, like so. And if I send another signal, it'll go up again, like so. And it'll go up until we hit this point, which is called maximal tension. Okay? So what you see is that, notice that in each little wave for each individual twitch, it becomes additive by that much, right? So this sort of additive nature is something that we call wave summation. Wave summation. Now eventually you'll get to this maximal tension, which is also what we refer to as tetany. Okay? And so this is what we refer to as unfused tetanus. And you'll see why here in a minute. All right, so now let's go back. Let's take this back to a relaxed state. Right? So what happens? Send a signal. It goes up. But what happens if I send a signal before it hits that stage of relaxation? Right? I'll get another wave, send another signal before it even starts to relax. What's going to happen is I get this straight beeline up to that maximal tension, and then it will relax like so. This is what we refer to as fused tetanus. Right? So unfused tetanus is where you get the sort of separation between the individual waves. Your fused tetanus is where you have zero separation in your waves, and it's one linear movement towards maximal tension. So when we move, we make these nice fluid movements, right? Time after time, we're operating under the conditions of fused tetanus because we don't move and make it look like we're under a sort of strobe light. Okay, so if you want to see this, right, you can watch the video that I have where it shows what happens at a frequency of signals at 1 hertz versus 5 hertz versus 10 hertz, because you can see individual muscle twitches, unfused tetanus, and fused tetanus.